Hey everyone, thanks Sam for sharing the screen. So today we're going to talk to you a little bit about Clarospec, which is a technology system based around hyperspec for measuring meat quality. So just before I launch in, do you want to go to the next slide, Sam? So the three of us are going to talk to you today. Myself, I'm in meat science with few years experience in R&D in the meat industry. Mos, who's a mechatronics engineer and has a lot of experience in deployment of technologies in industry, and Sam, who's into physics and imaging, segmentation, modeling, etc. So next slide. So the motivation for our research around measuring meat product quality was, particularly with lamb, we have 70% of New Zealand lamb that's suboptimal in terms of consumer expectation around eating experience, and 30% of the optimal product is unidentifiable or undifferentiated. So we're busy producing lamb, getting better and better at it in New Zealand, but we're actually missing the mark a little bit and leaving, I would say, money on the table from an export perspective because we're not being particularly consumer orientated around that eating experience. So our research into this topic goes back many years, starting with NIR and other things, and I've worked with Ian and Reddy on that some time ago now. But the most recent iteration was an MB Endeavour program where it was just recently completed last September, where we actually went through a whole range of technologies and arrived at hyperspectral as our preferred modality, and then used modeling approaches to predict the eating quality. And the outcome, what we're trying to achieve, is really doing more of a lamb by design type approach, where we're hoping to use this information to help the value chain or supply chain tailor production to meet those consumer expectations, to increase consumer confidence and increase value. So next slide, please. So what we're really about is enabling a consumer-informed supply chain through a data-driven decision process focused on eating quality, which means consumers can have some confidence in the quality of the product they purchase. It could be through like a quality tick or something like that. Processors know which farms produce the best quality meat, and farmers know what genetics produce the best quality and therefore the best return. So we're literally trying to link those consumer outcomes at the right-hand side of your screen, back down through the supply chain to the breeders, to the farmers, etc. So, and we're installed some hardware in the meat processing factory to do that. So next slide, please. So I'm going to hand over to my colleague Mos Sharifi now to discuss this slide.
Good morning, everyone. So our technology is basically a combination of hardware, software, and prediction equations to generate the meat quality attributes in this case. In terms of the hardware, at the heart of it, we have a hyperspectral imaging camera, and basically there is another industrial PC edge computing system that runs a software that we have developed. And what it does is basically the software does all the data capturing, data processing, and putting that process data into the prediction equations to generate the data. So it's a kind of a unique combination of these three components. In terms of the hardware, we have different deployment scenarios to provide us with maximum flexibility when it comes to measuring quality of fresh produce. We do have line scan system. In this case, you can see in the picture, this is Clarospec, the tunnel version, if you call it, which incorporates a conveyor belt, meat passing through it. And we do have a vertical scanning or a side scanning to, again, provide that flexibility to scan the fresh produce at different configuration. We are working on another deployment, which incorporates a mirror system and that is when you have your basically fresh produce static, and with the mirror you can basically get a snapshot of the cut surface of that fresh produce. So again, in this picture, what you see is a standalone Clarospec tunnel version. But when it comes to the industry deployments, there are whole heaps of other parameters that come into play, make it more challenging. And in this case, different meat plants, they have different processing systems or combination of components in the line. But what we're trying to do is to work closely with the end users, basically, and also have the system as modular as possible so it can be integrated into the line. Yep, so that's from me. Thank you.
Thanks, Mos. So I'm going to take everyone through our process of generating models and doing our data analysis. I'm sure that a lot of this is going to be super familiar to a lot of you, but yeah, we have our hyperspectral imaging system, which collects our images. We do a correction for the lighting, not as sophisticated as the parameters we saw in the first talk, but that seems to work well enough for our data. We collect an image. These have many wavelengths. And so we do a bit of processing on there to turn it into something that our statistical models can work with. And we match up the data that we get from those images with our reference data. The reference data that we are particularly interested in for meat is, oh, here, sorry. An example of the data that we're looking at is here's a lamb loin. We are interested in just segmenting out the loin and doing some processing on there. And so that's the first step in our processing once our data has been collected. Currently, we do a feature extraction on that, which is just to average all the pixels in that region of interest to get a single spectra. And then we do currently statistical modeling, PLSR, to predict IMF and pH. Very similar to Ian's talk is that we are actively looking at deep learning approaches to each one of these steps as well. Some of them, I'm not sure whether we'll get a big benefit from deep learning, but one that we're really interested in is better segmentation of the different components of the sample using deep learning. Some really interesting results from that. There's another. And I'm interested to hear what other people think in this as well. There's some indication that deep learning models are better for highly skewed data, prediction of highly skewed data, which we have some examples of coming up. So we're also, like Mos mentioned, we have a couple of different configurations of that of the Clarospec. And so we're looking at lamb, but also salmon. We have a project with salmon and beef as well. So the system's dynamic enough to be applied to a range of different products. The parameters that we're interested in particularly are intramuscular fat and pH. So both of these have high, good indicators of the consumer eating experience. So higher intramuscular fat is more tasty meat, which is good. And high pH is an indicator that your animals were stressed at the point of slaughter, and that results in tougher meat. And so we are interested in predicting both of these traits using hyperspectral imaging. But to build our models, we need reference measurements. And here we start to get into the complicated part, I suppose. IMF has quite a nice distribution, and so that's good for modeling. However, the collecting reference measurements is very expensive, and it also takes time. And so the samples have to be freeze dried and homogenized, and then chemical analysis done. And that is, yeah, that's an issue for just collecting enough data to be representative to build models. Fortunately, we have this visual marble score, which is basically just where someone looks at the sample and says whether it's high and low fat, as a proxy for IMF for subsampling. pH is cheap to collect, but it still requires a person to basically stab your meat with a probe, which is not desirable. And in this case, nearly all of the meat is within a very small range of pH, but high pH samples are what we're looking for. And so you get results that are, you get an underlying distribution that is very skewed, which makes it difficult to build models. So here's an example from a recent trial of that we conducted at Agresearch with around 780 samples of lamb loin, and just an example of the pH that we got from all of those samples. Nearly everything is between 5.6 to 5.9, but these ones that are getting up into the high pHs are what we're really looking to identify. And PLSR is not so good at picking those out, and so we're interested in a deep learning approach to do a better classification of those. For the marble score, we had the wet chemistry results for this trial haven't actually come in yet, but the marble score that we got, you can see most of the data is marble score 3, which roughly equates to between 2 and 3% fat. But because we have this measurement, this estimate collected on the day, we can subsample those to get an almost flat distribution across the range of IMF for building models. And so Agresearch has been doing this for a long time, doing a trial, getting a data set, splitting calibration, validation, building a model on that 120 odd subsample, and then predicting the IMF on the remainder, and using that data for writing papers and providing breeding values to farmers and that sort of thing. We are, now that we're in the phase of actually deploying our device in plant, we're really moving away from this idea of doing a single trial and building a model that's specific for that trial. We want something that is quite robust across time and years. So I just quickly go through some papers that we published last year and the year before about how we are dealing with having a model that is deployed, checking the performance of that model against real world data as it starts to come in, and then what do we do with the model? In this example, this paper, we did not a very sophisticated approach, which was just as soon as that new data came in, we recalculate the PLSR model, produce an updated model, and then check the performance against the next years or the next trial, the month later or whatever. In this case, it was an interesting process to go through, but honestly, the updated model wasn't significantly better than the model we got just from collecting the first year from 2014, collecting just that data, building one robust model and then predicting all the subsequent years. A more interesting approach, and which is where we've leaned to, is to basically collect as much data as possible, but from a range of different sources. So here we had a collection of 26 independent near-infrared trials, but these were different because the sampling protocol was different across the years. The devices were different. We used 4 devices across those years. And this was a really interesting exercise to go through to to learn about device transferability and model transferability, and how using multiple devices can actually build quite a robust model.
Just 1 minute to go.
Yep, thank you. So anyway, final couple of slides. This device has been deployed in plant, and it's been collecting data, and the first round of wet chemistry results have just arrived, and basically we're very happy with the performance that we're getting. We have two models that the client, I suppose, can choose from. One was built using beef and lamb, and another was built just using lamb. And yeah, they're both biased, and we expect that because these models were generated on a completely independent data set from what we are looking at. And so there's a big push now to develop methods to not to do a full recalibration, because we showed in that paper that that's not that useful, but a tweak to the model as you slowly start to get data coming in, I think is going to be the way to go moving forward. And we have some initial results are looking good with that, but need to cross some I's and other way around. Cross some T's and dot some I's before moving on. So I hand back to Cameron for just the last slide.
Yep, thanks, Sam and Mos. So just briefly, our next steps are build and deploy a commercial prototype that gives us the real world case studies, maintains our momentum, and enables us to validate the pricing and commercial models, etc. And then we're looking to actually get this out into the real world, either through a spin out or some other mechanism to get it beyond the research stage. So really love to hear from anyone who's got some insights either on the data side or on that commercial side about things that would help us on our way. So thanks very much.